Everyone. I am Dr. Nisreen from Georgia University of Science and Technology. I will be talking today about the effect of drought stress on wheat, triticum, durum, growth, and metabolism inside from basically GABA Shunt reactive oxygen species and the dehydrin gene expression. First, I would like to thank the GPMP 2022 family actually for allowing me to be actually the moderator, then to have my presentation actually in the meeting. So let me start first. I will be basically going through an introduction a little bit about the just please. Okay. So I will go about just a brief introduction about the wheat, biotic antibiotic stress, then I will talk a little bit about the drought stress, and moving to GABA Shunt pathway, which is actually the core for this research, and then the relationship between the GABA Shunt pathway, the tolerance of those with the crop, and the expression actually of the GAD gene and the dehydrin gene. Then let's talk about wheat. Wheat actually, that's used in this study, it's a Triticum durum, which is, of course, it's a monocotyledonous crop from the Poaceae family. It's the second cultivated species of wheat after basically the common wheat and Triticum estivum, and it represents about 5%, around 8% of the global wheat production. And of course, wheat, like any other plants, it's subjected actually to environmental factors. And in Jordan, my country, we are facing the problem with the droughts, and we're looking up for crops, basically, especially like it, that most people they depend on it as nutrition, or the main actually, let's say, part of the menu. That would be a response, or that would be adapted to drought stress. Now, plant stress actually, it's classified, as you all know, as biotic and abiotic stress, and mostly biotic stress is occurred by other living organisms like viruses, bacteria, and insects. And the abiotic stress, precisely, it's caused by physical and chemical environmental conditions that can alter the growth and the production and also the productivity and the yield of the crop, such as drought, temperature, involved plant, heat and cold, salinity, and oxidative stress. So drought stress, which is the core actually focus about this study, usually is defined as a low level of water availability in soil, which trigger morphological, physiological, and molecular modifications in a plant tissue to respond to low water availability. And reduced water availability and scarcity in the soil, it results in the accumulation of salt, and toxicity actually fires around the roots and causing a second kind of stress, which is the osmotic stress and the salt toxicity. Now, drought stress as an abiotic stress, it reduces water potential and trigger pressure in plants. It's induced tomato closure. It decreases photosynthesis and generation of reactive oxygen species and causes an oxidative stress. It's altered the membrane stability, causing membrane damage, and decrease the chlorophyll production, which is responsible basically for photosynthesis, that's synthesizing actually the nutrients for the plant, and also it will cause ion uptake and transportation that has a negative impact actually in other metabolic processes inside the plants. And also drought stress, it results as imbalance between the production of the reactive species actually and also the availability in the work of the antioxidant protection. All of this, it will affect the production or the yield actually of this crop. That's why we choose this abiotic stress to work on. Now, drought stress also, plant, it's a living organism, and precisely it will respond to drought stress by certain strategies. It's enhanced root growth, and it's decreased water loss, by reduced epidermal or critical action actually, depression of radiation, absorption, and decreases actually the evaporative area. And also inside the plant, at the molecular level, a group of genes. They will be activated and they will be expressed to enable the plant to respond to drought stress. Now, the core study, the core actually talk about, or the core section of this study, which is the GABA shunt pathway. I've been working on GABA shunt pathway actually for about 13 years since I finished my PhD in 2008. And GABA shunt pathway, it's a pathway between the cytosol and the mitochondrial matrix. In a plant, GABA perform as a multiple metabolic and physiological function under stress actually, and also non-stress conditions. It's a neurotransmitter in animals, and it's found in bacteria, but in a plant, it's linked as endogenous signaling in plant growth and development, precisely since it serves as a carbon and nitrogen source. So it's a play an important role in carbon and nitrogen simulation, especially under stress. And it's metabolized actually by critical pathway. That's why this is GABA shunt pathway, because starts in the cytosol and then it will be shutting inside the mitochondrial matrix. This is the GABA shunt pathway. Now, GABA actually it's produced from the glutamate by decarboxylated the glutamate by an enzyme called the glutamate decarboxylase, which is a calcium calmodulin dependent enzyme, the critical enzyme actually that initiated the metabolic pathway itself. And then GABA, it will be transported from the cytosol to the mitochondrial matrix, which enter actually what's known as GABA shunt. And GABA shunt, it's feeding actually an intermediate, which is the succinate, after GABA converted to succinate semialdehyde, and then succinate semialdehyde by succinate semialdehyde dehydrogenase, it's converted to succinate and entering the Krebs cycle. And of course, this will be involved in the stability of carbon nitrogen source in plants. So what I will be talking about here, actually focusing, we're going to talk about the level of GABA that will be accumulated in response to drought stress, and also the two other amino acids, but they are recognized as GABA metabolite, the GABA shunt pathway metabolite, which is the alanine and the pyruvate. Because GABA inside the mitochondrial matrix, it's converted to succinate semialdehyde by an enzyme called GABA transaminase. So for who will be receiving the amino group from GABA, either alpha-ketoglutarate or actually converted to glutamate, or pyruvate is converted to alanine. So we're gonna measure, in this study, we measure GABA level, we measure the glutamate level, and also we measure the alanine level. Now, GABA as an basically a key intermediate, or also a signaling molecule, there is a lot of obscure things about it. It's not 100% known, but its actually, it's involved in metabolism, osmotic adjustment, it's enhanced membrane stability. As a carbon and nitrogen source, actually, it will stabilize the energy metabolism. All those group of basically pathways that GABA and correctly could be entered with, it will enhance the abiotic stress tolerance. Now, what I was interested to actually is to see the relationship between the accumulation of GABA under drought stress and the expression of dehydrin genes. We focus actually on two dehydrin genes. And as you all know, dehydrin genes, they are upregulated during the drought stress, and they are related to oxalic acid regulated pathway. They encode the protein like latent perogenesis abundant protein. And we're going to talk about two genes actually in this study and their expression: the dehydrin, which is the type of the latent protein gene that produce actually a response to oxalic acid basically release and pulse, and also the wheat called regulated gene 410, which is also another dehydrin gene that's increased around the plasma membrane, and it's upregulated actually by not just cold, it's also by a lot of it, also by dehydration, and also it will be activated the exhibition response to ADA treatment or oxalic acid treatment and the salt stress. So as you see, dehydrins, they play a very important role to make the plant adapted, not just to drought stress actually, but also to another abiotic stress. So the objective of this study was to investigate the effect of drought treatments in durum wheat. Actually, we focused on 4 cultivars that they are cultivated in Jordan, and they're seedable and also at the seedling stage in terms of seed germination, seedling growth. We looked into metabolism to see the effect of GABA shunt metabolism. We measured the GABA shunt metabolites, GABA, alanine, and glutamate, and we measured the level of MDA, which is as a reflector actually for the reactive oxygen species. And then we extracted total RNA from seed and also post-germination, actually 8 days post-germination, and also from the seedlings, and detect the expression of GAD, the glutamate decarboxylase, and the other dehydrin gene. Of course, the main, basically, important point why we did this, or we looked actually for this parameter, is just to investigate the rule, the functional rule for GABA shunt metabolite, and also related with reactive oxygen species, and then scavenging and connecting it with the expression of glutamate decarboxylase, dehydrin, benzoate. The main point, or the output actually, this is to maintain the metabolic adaptation, to see how much actually the expression for those genes and the accumulation of those metabolite involving adaptation, stability, under all drought treatments that were used actually in this study at the seed stage and at the seedling stage. So in this study, we use 4 cultivars. They are Triticum durum, cultivars, Quraan 75, Sad 65 for Shaman. Those are Jordanian, wheat cultivars from the Triticum durum. They were obtained from the Jordan National Agricultural Research Center, and they were harvested actually just the year where the study was done. Of course, first we sterilized the seed with about 6% volume by volume soluble hydrochloride, and we flushed them with distilled water, and then we fed them for seed germination, and then we go for the seedling stage. So basically, first we looked up for the sensitivity of seed to drought treatment, and the wheat seed were planted into filter paper as artificial growth surface. We saturate them with water in a drought manner system according to water saturation level, 80%, which is full saturation, 50% moderate, and 20% low water availability, and for separately. And then we incubate them for 8 days at 25 degrees for separately for each one of those cultivars. And then we looked at the physiological growth parameters. Basically the experiment, we run 2 experiments. We looked up actually, we make the cultivars growing for about, when they reach about 10 to 12 centimeter long, and then a group subjected to 7 days water stress or drought stress, and the other group for 14 days separately. So we have 2 groups. After 7 days, we measure the metabolic assessment and the gene expression. Now, in metabolite extraction, after 8 days post germination, also after 7 days drought treatment, and after 14 days drought treatment, we extracted actually the GABA according to Zangenbaum 1997. GABA as a metabolite actually, or the metabolite supernatant, we used to determine the GABA level, and alanine, and also the glutamate. Of course, GABA was determined by using the nicotinamide adenine dinucleotide phosphate standard care, but for alanine and glutamate, alanine using alanine dehydrogenase, and glutamate with glutamate dehydrogenase using the nicotinamide 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 diadenine as standard care. So we measured GABA level, we measured the glutamate level, and we measured actually alanine level. As I said before, let me just take you to remind you why I focus actually on those 3 metabolites, because we are measuring GABA as its amount, and then we're linking it how it will be converted either to alanine actually, or to glutamate inside the mitochondrial matrix. And then after that, we did measure the reactive oxygen species, or basically the level of MDA, again 8 days post germination, and for 1 week and 2 week drought treatment tissue just above the ground was harvested, and we basically using the APCAM lipid peroxidation MDA assay, calorimetric kit basically, and we measured the lipid peroxidation as malonaldehyde equivalent as was determined actually for the lipid peroxidation assay. Then the same, the seed actually 8 days post germination, and also after 1 week and 2 week drought treatment, we harvested the shoot tissue, and we extract the total RNA using the RNA easy plant extraction kit actually from Intron Biotechnology, Korea. And then we follow just the manufacturer instruction to look up for the next step, which is determining the expression of glutamate decarboxylase and the dehydrin genes. Of course, GAD messenger RNA expression, it was done by the reverse transcriptase. We use one step RT-PCR premix kits, and then we calculated the fold expression related actually to internal control, which is the 18S ribosomal or 18S actually RNA, using it as internal control. So for every treatment or for every parameter we measure it, we use 3 treatments. 8 days post germination, after 7 days of subjecting the seedlings to drought stress, and also after the 14 days. And the data was calculated as block 2-fold in abundance actually for the GAD expression, also for the abundance of the dehydrin genes, the DHN and the W4 genes basically, and the same internal control was used. But of course, data was experimentally analyzed. We did it as a completely randomized design, and we used one-way analysis of variance to analyze each actually parameter under all treatment individually. Let me just go for the result and discussion. First, for the seed germination. Seed germination of durum from this table you can see for all the 4 cultivars, they did perfectly well actually at 80% water holding capacity or saturation level, and no difference, as you can see, not that much actually compared between 80% and 50%, but you can see at 20% all 4 cultivars they failed to germinate. So there were no germination actually at a 20% water saturation level, which has a very severe actually impact on seed germination. And we can see we have actually a positive correlation as we go with low decreasing the water availability, the germination percentage was decreased. So in this study, drought stress significantly, as you noticed from the previous table, reduced seed germination in durum for all cultivars. And the 80% water holding capacity actually providing the enough water for the seed to successfully germinate, providing enough water for physiological, biochemical, and molecular process to be activated during seed germination stage. And when water holding capacity decreased to 20%, the absorption of water seed was decreased or stopped, which basically prevents their successful passing germination stage and affecting their metabolic and enzymatic activity that will be required actually and actually essential for seed germination. And they suffer actually from accumulation of salt stress and causing osmotic stress and ion toxicity to seed when the water availability decreased to 20%. Now, seedling growth for durum wheat under drought stress for 7 days post treatment. So basically, we looked up for plant weight, fresh weight, dry mass, and the water content after, of course, that 7 days. And you can see as the water saturation level decreased, the basically plant height and it's decreased. Also the same for the fresh weight, dry mass, and the water content. The same scenario actually it was noticed basically for when the seedlings was subjected to 4 days post drought treatment. So this study, I just want to mention one thing. All the 4 cultivars actually, there's certain variability between the cultivars. They are responding to drought stress differently, but all of them they showed a positive correlation with as you decrease the water level, the physiological growth they were decreased, and also all of the data they were significant actually under all water stress treatments after 7 days or after 14 days. This is just a representative picture you can see for the seedlings when we just harvest them basically. That will be taken to calculate to measure basically the growth parameters. So our study indicated that different treatment of water stress it will be leading to decline in the fresh weight, to dry mass, which was the most determinant of, let's say, the descriptive physiological parameters. They are very informative. as well as water content for all durum wheat cultivars. They were using this system. Also, the reduction in the physiological growth parameter of durum wheat were more significant actually after 14 days. It is expected because after 2 weeks, the plant is suffered and start to survive actually for by comparing to 7 days post-drought treatment. Now, under continuous severe water stress, what's happened actually? Several drought strategies that could be used to escape or to adapt to the plant survival under the drought stress. That's why we looked up actually for the metabolic assessment and the ROS level and the expression actually of GAD and the Dehydrin gene. Of course, the negative impact of water stress on the shoot growth can be explained as an adaptation to water storage by limiting the rate of transpiration. So decreased growth, so less water, less surface area available actually for transpiration. So this is a strategy to escaping this severe drought, especially after 14 days post-treatment, post-drought treatment. Then for those seedlings actually after 7 days, and 14 days, we took the shoot and then we measured the amount of GABA shunt metabolites after 8 days of germination stage and also after 14 days. And you can see as the water level decreases, or the saturation level decreases, the amount of GABA itself as a metabolite is increased. You can see from the negative correlation here. Now GABA increase, the abundance of GABA was more, with small abundance actually of alanine and glutamate that will be explained later. The same trend actually it was showing in the seedling stage. So we measure it 8 days post germination and also after 14 days and after 7 days for 1 week and 2 weeks actually for all the 4 cultivars at this post-drought stress. Now, from this study, from this actually measuring this parameter, we can see that GABA abundance increased actually more was associated with small, okay, but also significant increase in alanine and glutamate level in all durum wheat cultivar under all drought treatments. And the shortage in water availability during seed germination and seedling stage growth actually was also significant. It's contributed to this high level of significant elevation in GABA content to reduce the durum wheat damage, because we said GABA contributed to membrane stability, organized nitrogen metabolism, actually amino acid metabolism, biosynthesis, contributed to stability of carbon and nitrogen metabolism as it's actually influx through Krebs cycle, and also regulated the wheat growth under drought stress. Now, GABA level was higher during post-seed germination comparing 7 days and 14 days seedling treatment, and this because significantly the critical step or the critical stage for plant growth is during seed germination. So seed germination is the most important and it's the most sensitive stage during the plant growth and development. So the elevation of GABA is just to enable seed to adapt this low water availability and to maintain or striving actually for successful germination, especially under 20% water holding capacity. Then we looked at the accumulation of reactive oxygen species also after 8 days post-germination and also after 7 days and 14 days seedling stage drought treatment. As you see, the highest level actually of MDA it was detected at the 20% saturation level, with of course a negative correlation as you can see from this table. As the water availability decreased, actually the amount of MDA or the neuronal divide accumulation it was increased in all dormants. The same basically during seedling stage after 7 days and 14 days, we noticed actually the same trend of accumulation of MDA after 14 days of drought treatment and after actually 7 days of drought treatment. Now, MDA content, why we measure MDA content, it was used as an indicator of oxidative damage. In cell membrane, especially in durum wheat, actually that would be induced by drought stress, because drought stress is associated with accumulation of ROS, and the result indicated that severe water availability, especially at the 20% water holding capacity, it actually primed the oxidative damage to cellular component and elevated the ROS production actually in all durum cultivars that were used in this study, Qurani, XF65, on-pace actually, and Shunt. And the accumulation of NDA level was higher in seed post-germination, as we noticed actually from the table comparing to the seedling stage. This could be justified due to critical sensitivity, as I said, where seed germination, sensitivity of seed germination to low water availability. And also the severe water concentration or the low water availability, the 20% saturation level, it's actually with the increase of time, especially after 14 days, it's caused severe damage to the cellular components and may lead to death actually in all durum cultivars. Now, sensitivity of different durum cultivars, it was basically correlated with the accumulation of NDA level under all drought stress or drought treatment in the system. And the degree of tolerance in durum wheat, it's connected actually with ROS scavenging system and the availability of the activating antioxidant enzyme. We did not actually measure them in this study, but reflecting that the ROS availability, it will be connected with the activation of antioxidant enzyme. Of course, it's associated with the activation of GABA shunt pathway and the production of GABA in seed by seeding the response actually to water availability. As I said in my presentation, GABA, it could work actually as a scavenging agent for ROS accumulation. Then we looked at the expression of GAD, okay, also at 8 days post-germination and 8 days post-germination, and at the seedling stage, 7 days post-treatment and 14 days post-treatment. As you can see, the highest expression level or look-to-fold expression actually, it was observed 14 days post-treatment, the seedling after 14 days, which means that the activation or the action of the GABA shunt pathway, it's go actually, let's say, it will be increased as we go with time, that will be subjected to plant to drought stress. So if you look at this plot, I just make this plot to show the amount of the expression of GAD in all the 4 cultivars. This is seed 8 post-germination, and here in seedlings 7 days post-treatment and seedlings 14 days post-treatment. As you can see, at 15% and 20% total holding capacity, basically the highest accumulation or the highest expression or abundance of the GAD was basically measured. So low water availability and the prolonged exposure of durum cultivars to drought stress, it increased the GAD expression and activated GAD mediated the conversion of glutamate basically to GABA and activated the action or the enhancement of GABA shunt pathway in the plant, especially as you can see here at the seedling growth stage. And also the activation of GABA shunts through the upregulation actually of GAD expression, enhanced its expression and production, it was correlated with the GABA accumulation to maintain the carbon-nitrogen balance, also the metabolism of amino acid and the carbohydrate stability, and also the regulation of the growth, and of course maintaining the cellular pH homeostasis during or under drought stress. Now the level of GABA metabolite, as we noticed, it was not high at the seedling stage comparing actually the 8 days post-germination. This can be explained that GABA, it was consumed. It to be indicated the consumption of GABA as a metabolite in carbohydrate and amino acid synthesis and protein metabolism or metabolic homeostasis. Of course, to be consumed for osmolyte synthesis to maintain the growth for durum wheat, especially under all those drought stress treatments. Then we looked up for the dehydrin genes, dehydrin actually, and the W4 gene to see the expression for those genes at all those 3 different stages actually planted, but 8 days post-germination and also 7 days post-treatment for the seed with a 14 days treatment for the seedling. And we noticed increase actually in the expression of the dehydrin gene, as you will see here, but actually, in the contrary, the GAD, the expression of the dehydrin, it was higher, and the abundance of the dehydrin gene during the germination stage higher than the seedling stage. So the level of abundance actually of the dehydrin messenger RNA post basically germination stage was higher comparing to the seedling stage, and this might be indicated that the function for those clear protein They work in the later stage of seed germination. That's why their expression actually is increased. And during the perigenesis and development to stabilize membrane, cereal molecules, and also activated enzymes required for successful seed germination and response to water availability. And increase in the dehydrin gene expression, it was also associated with ROS scavenging and less or lower level of MDA accumulation basically during this stage, because they act as a molecular chaperones and osmotic adjustment, brain stability, and also they work as a functional dynamics in response to ground stress. So you can see from this plot the highest accumulation actually, or abundance for dehydrin gene, which was at the 20% saturation level of drought symptoms. Now, we looked also for another dehydrin gene expression, which is W414, basically in also at 8 days post germination, and also in seedlings, 7 days post germination and 14 days post treatment. And we noticed from this plot here the same trend, okay, but in a lower profile comparing to the dehydrin, and the highest significant increase, it was also the same, like dehydrin after 8 days post germination, like the dehydrin gene. But if you notice from the previous slides here, we noticed that dehydrin actually as a protein, its gene increased its expression higher, comparing actually with the W410. And this can be explained, okay, that both of those genes, they are encoding the dehydrin protein, the late embryogenesis abundant protein, that they are playing a very important role during seed germination, especially under drought stress. So our result actually collectively found a significant increase in the W4 expression during post-germination, which suggested that W4 messenger RNA transcript abundance associated with the significant role of this gene, the W414 dehydrin gene in structural stabilization, which act actually a chaperone-like properties, protein and thus protected various nuclear and cytoplasmic macromolecules from coagulation during dehydration, low water availability. And dehydrin and W4 messenger RNA transcript also significantly increased with increase in the drought stress. We noticed at the 20% saturation level, they were the maximum abundance actually for those gene expression. And both of those genes actually, they play a critical role in adaptation to water stress and their overexpression. And it's proved to be a successful or useful, let's say, indicator to look up actually for plant adaptation to drought stress. So in conclusion from this study actually, drought stress has a negative effect on growth and metabolism in durum meat, all the four actually cultivars that we use in this study, of course with certain variability between them. Reduction in water content has a negative effect on seed germination in durum meat, which might be resulted from reducing water absorption, water availability for the seed during germination, and also reducing seedling vigor, are also affecting metabolic and enzymatic activity that would be required for successful seed germination, that lead actually to accumulation of iron and causing osmotic stress due to low water availability and increasing the salt stress, or a second effect of salt stress and increasing the salt concentration and causing salt toxicity. During drought stress, the stress itself significantly decreases seedling height, fresh weight, which are the growth parameters actually, as well as water content in plants. Now we know as water content actually it will be decreased, it will be causing more negative, more stressful impact during seed germination, and this will be causing extra water loss, and then that will be affecting the seedling, the seed germination, also the seedling survival. Drought stress during seed germination and seedling growth stages, it's also caused significant inhibition and activation the GABA receptor metabolic intermediate to tricarboxylic acid cycle, the Krebs cycle, which is the core cycle for carbohydrate metabolism plant, to maintain metabolic stability and metabolic homeostasis to control the osmotic effect and regulated the wheat growth actually under drought stress. And the level for the GABA metabolite, precisely going high level of GABA, and also significant increase in anaglutamate, was lower in basically during post-germination, comparing actually with the seedling stage. It's indicated that during seedling stage, the GABA, it will be consumed to be involved in protein metabolism, osmolyte synthesis, and maintaining the growth response to drought deficit. So it will be using its structural component, it will be used to synthesize osmolyte and also to stabilize the metabolism during seedling stage, actually comparing to 8 days post germination. And durum seedlings had significant increase also in MDA content. And MDA level, as we noticed, it was higher in post germination comparing to the seedling stage, due to critical sensitivity actually for seed germination to low water availability and the scarcity of water during seed to achieve successful germination. So the take-home message actually from this study, the degree of tolerance in durum wheat, it might be connected actually with the ROS level. And also it might be connected with the action of antioxidant enzyme that was also associated with the activation of GABA shunt way and the GABA shunt pathway and the accumulation, the production of GABA against drought stress. And the low water availability and especially prolonged exposure of those 4 durum cultivars to drought stress enhanced or activated the expression of the GAD glutamate decarboxylase gene, which is the key gene coded for glutamate decarboxylase enzyme that converted the glutamate to GABA, especially basically during the seedling growth stage. And the activation of GABA shunt through upregulation actually of the GAD expression, it was done to maintain the nitrogen carbon metabolism, of course stabilizing the mitochondrial activity, energy production, and the regulation actually of growth and cellular pH under drought stress. And also we noticed as as GAD or the activation of GABA shunt pathway, the dehydrin gene, especially dehydrin and the mucor that were measured in this study, they also upregulated during post-germination stage comparing to seedling stage, on the contrary of the GAD, to maintain, of course, the membrane protection because they are work actually as their protein and they work actually as bioprotectants for enzymes and they prevent the ROS, they act as an antioxidant agent for ROS accumulation in response to drought stress. Of course, understanding of gene expression, it will lead actually to strict control of physiological and biochemical responses actually of not just durum wheat, actually other cultivars, especially enable to express not just necessarily to drought stress in plants. And overproduction of the dehydrins, especially dehydrin that we were using this study, they are essential to improving the drought stress tolerance in durum wheats. And our data clearly showed that different wheat cultivar respond differently. Not all the cultivar actually they respond in the same way for basically in terms to drought stress, they respond differently, but all of them actually they show the drought stress showed negative impact actually in their growth during germination, actually during seedling stage. And further investigation also for of course to elucidate the signaling to understand the connection between the GABA shunt pathway and the expression of the dehydrin and the dehydrin action, it will be important and required actually for especially under drought stress. I would like actually to thank our university, Jordan University of Science and Technology, the Division of Research for funding this research. This research is funding by number 442, 2020. And this work actually it was in collaboration with my colleague, Professor Nizar Samara from the plant production department, and also with my work with my master's students, Ayush. Thank you so much actually for listening to my presentation. And I will be ready if you have any question. If you like, you could email me or post your question on the chat box, and I will be ready to basically respond to it. Thank you. I will stop the sharing. So if you have any question, I will be ready to take your question.